వద్దు.
నందు
ఇంకా
ఏమందు
రావట్లే కాల్.
అవునా?
మీటింగ్ అవ్వదు ఇంకా రావట్లే. ఆమె మార్నింగ్ అనుకోని మార్నింగ్ జాయిన్ అయిందా ఏంటి?
అవునా, నేను కాల్ చేశాను వాళ్ళకి
కానీ
ఒకసారి.
వాళ్ళకి వాళ్ళు చెప్తారు కదా, వాళ్ళు కన్సల్టెన్సీ వాళ్ళు ఒకవేళ
వాళ్ళు కన్ఫర్మ్ చేసి ఆల్రెడీ టైం 7 అని.
అదే కదా, నేను కరెక్ట్ అవ్వట్టు ఉండాలి కాసేపు.
ఒక 5 minutes ఆగు. 7:10 కి వరకు రాకపోతే చెప్పు, నాకు పింగ్ చేయ, నాకు కాల్ చేసి నేను వాళ్ళకి కాల్ చేస్తాను.
సరే, ఓకే. హలో. 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 హలో.
Am I audible now, Nandini?
Yes, yes, I can hear you, ma'am. Yes.
Okay.
Yeah. How are you, ma'am? How are you?
Thank you. Yeah. Yesterday's notes, I'll send you today, Nandini. I
Yeah,
didn't get chance
sure.
yesterday.
Okay, sure, ma'am. Thank you.
This conference will now be recorded.
As last class, we discussed with
Mhm.
the general process of clinical studies
Yes, okay.
and the role of CTM in the process of clinical studies, right?
Yes, yes.
Right. We'll see preclinical studies first.
Okay, sure.
Now, so that means that process that I'm going to elaborate
Okay.
is the preclinical. So, what is the main objective of preclinical?
It's on the clinical research on animals, clinical trials first. They
Mm,
do they do conduct research on animals first.
mm,
Yeah.
right. What is the main objective of preclinical?
Mm, to be safe and effective, it's if it's a drug released to the approved approval.
Mm,
Yeah.
pharmacology mainly.
Okay. Yeah.
Pharmacokinetic,
Dynamics.
pharmacodynamics.
Okay.
So, yeah, what is pharmacokinetics and what is pharmacodynamics?
Yeah. Pharmacokinetics means what body does to the drug, and pharmacodynamics means what
Mhm.
drug does to the body.
Mm.
Yeah.
It
Basically
is
it's a
all.
drug movement
It's,
and okay.
right, drug movement
Yeah.
in the body.
Yes.
Right. So, movement in the body.
Yeah.
Right. So, we even go for toxicology also.
Okay. Mm. Yes.
Okay. So, genotoxicity, reproductive toxicity, gastrogenicity, acute toxicity.
Okay.
This is all toxicity studies. So, before the new substance used as a
Medicine.
medicinal product,
Okay.
before.
Hello. 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 Yes.
Yeah.
Okay.
Okay, so before the new substance used as a medicinal product,
Okay.
it has to be tested for safety and efficacy
Mhm.
in animals before it's used in humans.
Okay. Hello.
So,
Yes. Hello.
today go to meeting is acting weird.
Okay. Yeah.
Okay.
It's like suddenly. Okay.
Suddenly it got disconnected. Yeah.
Okay.
Generally, it won't happen like this.
Mm-hmm.
So let's see.
Okay.
So the main goal of, you can see this testing is called preclinical.
Preclinical
In the
stage.
drug development, it is a stage of research, which will definitely begin before we start clinical studies.
Okay.
You can see the main goal of study. So mainly, as you know, pharmacokinetics and dynamics
Yes.
and toxicology.
Okay.
Here we have given 3 points. The first one is safety evaluation. Second one is what are all the parameters that are required for the
Monitoring. Okay.
safety parameters for the clinical monitoring.
Okay.
That means before we go to clinical studies, what are the parameters
Okay.
to assess the safety? That will be determined in the preclinical.
Okay.
Okay. And also identify the potential target organs.
Okay.
Potential target organs for the toxicity studies.
Okay.
Okay.
Yes.
So as you know, kinetics ADME, how the product is absorbed, how it is metabolized,
Yes.
how much
Distribution.
amount of drug is excreted,
Yeah.
yeah, how much amount of drug excreted, and how it is going to
Excretion.
be distributed.
Yeah.
Right. Along with the liberation also. Liberation also will be the part.
Yes. Okay.
Right. Generally, after screening, phase step before preclinical, we have a lot of steps actually.
Okay.
Before we start preclinical, we have a lot of steps.
Okay.
Like drug development process, it is called. Like screening,
Mm-hmm.
lead compound, lead identification, protein modeling, protein binding, these are all.
Okay.
After all these, before we come to preclinical, we will conduct the first step of clinical nothing.
Okay.
So what we do before we start preclinical, we'll do the screening part. Let's say if 10,000 molecules are screened, out of which only 250 molecules will be come to the phase.
Okay.
Which phase? Preclinical phase. Out
Preclinical
of
phase.
10,000, if 10,000 are, yeah, if 10,000 molecules are,
Conference will now be recorded. Be recorded. Be recorded. Be recorded.
so only 250 molecules will be selected for this phase.
Okay, preclinical phase. Okay.
Out of 250, only 4 to 5 will be selected for the clinical study after preclinical.
Oh, okay.
Then after clinical, out of 4 to 5, only one molecule will be released into market.
Oh my God, it's a very big process.
Right. So almost 10,000 molecules to we choose only one
One
molecule.
molecule. Okay.
So this is a process.
Okay.
That means while conducting preclinical or clinical, we will not only focus on just one product.
Okay.
Then before we start preclinical also we have to get approval by the regulatory authorities.
Okay. Before
So we,
preclinicals also and after preclinicals.
before preclinical, after preclinical means before we start clinical,
Okay.
before we start marketing.
Okay.
3 times we have to get approval.
Okay.
So regulatory authority must ensure,
Okay. It is safe to.
regulatory
Okay.
must ensure that the clinical trial conducted in safe, ethical manner
Yes.
and should give approval for those drugs which are found to be safe, effective. So this is pharmacology, you can see.
Okay.
Get it?
Yes, I got it. I got it.
Right. Dynamics also you know.
Yes.
Toxicology also, genotoxin means whether the product is mutagen, means whether it has any genetic mutations,
Okay.
whether the product has any effect on the reproductive organs,
Okay.
and toxicity, whether the drug is cancer causing.
Chronic conditions. Okay. Yes, got it.
Acute toxicity within the shortest possible time. After giving them the drug within the shortest possible time,
Okay.
will observe side effects. Okay.
Okay.
So in case if it is showing any time side effects, we'll stop there.
Okay.
Repeated dose studies, repeating the same dose administration, drug will be given.
Okay.
We'll understand the toxicological properties of, properties of the drug,
Yes.
toxicological profile of the drug.
Okay.
After completion of preclinical studies, there will be pre-formulation study. After pre-formulation, then formulation will be started.
Okay.
So what is pre-formulation?
It's either tablet or the capsules or liquid formation. The drug.
Mhm.
Yeah, it's
Right. After completion of these preclinical studies,
Okay.
what is the next step then? What is the next step?
Clinical trials.
Right.
Yeah. Preclinical
We
trials.
have to go for clinical trials.
Yes.
The next step is clinical trial.
Yes.
Testing the drug on human beings
Human beings.
to
Yes.
find safety and efficacy. Right. Before we start clinical trial, what will be the steps?
We need to approve the approval. We need to take the approval from investigational the investigation bureau. We need to get approved from IRB. Is it right?
IIT,
We need to get IRB, Institutional Review Board.
no. Nandini, approval from
Oh, IND.
F-
FDA. Okay.
Food Drug Administration. It is a regulatory authority in US.
Okay.
Submitting application called INDA.
Okay. Okay.
Okay, investigation new drug application will be submitted to the regulatory authorities in
Okay.
order to get the approval,
Okay.
investigational approval.
Okay.
Okay, let us discuss INDA. It is the application by which pharma companies get approval to conduct clinical studies. With this approval, we'll start uh, shipping the product.
Okay.
Start shipping the product across all the clinical sites.
Okay.
FDA will review that. Once you submit, then the INDA, FDA will review that. They will make sure that the patients will not be subjected to any unreasonable risk.
Okay.
If the application is clear, then the drug usually enter phase 1 clinical studies.
Okay.
What does it consist of and what is the title here? You can see 21 CFR Part 312.
Okay.
Okay. So what is CFR? Okay, tell me what is CRF?
Contract Research Organization. Contract Research Organization. Right.
The name that is CRO.
Oh, CRF case report form.
Hmm.
Okay.
CRF is a case report form. So CFR is different, CRF is different.
Yes. Okay. CFR means I don't know, ma'am.
Yeah, Code of Federal Regulations.
Okay.
General regulations across US, not only related to particular body system,
Okay.
but not only related to uh, the clinical research topic, not only related to clinical research, not only related to only food and drug.
Okay.
We have a lot of titles and lot of areas covered in that. So Title 1, General Provisions, 2, Grants and Permissions,
Okay.
3, President, 4, Accounts, like that. Okay,
Yes.
right. So we have 1 to 8 titles. Actually, we have 50 titles. different
Yeah.
titles that we have. Out of all the titles, title 21 is the title which deals with food and drug.
Okay.
Okay, you can see the list. Out of all the titles, 21 is the
21
title
is the
which
food
deals
and
with food.
drugs. Okay.
45, public welfare.
Okay.
Okay,
Yes. Okay.
right. In this, we have many parts, sections, subsections. Out of all the parts, part 312 will give you guidelines related to INDA, means how to submit, when to submit, how to, what to include and all will be given in INDA.
Okay.
Then, what to include in INDA? So, generally, tell me what do we include in INDA?
All regulations like food and drug welfare.
No, no. In the INDA, what do we include? Actually, when you're submitting INDA, we follow CFR guidelines 312.
Okay. Like safety regulations. Like.
Mhm. Whatever the data that we collected from the preclinical study, and what exactly that you're going to do with the human being.
Okay.
Both the data will be collected here.
Okay.
Animal physiology,
Physiology.
toxicology,
Chemistry.
animal pharmacology studies data, chemistry, manufacturing details, protocol that we are going to follow because FDA should know how you're going to conduct the clinical studies. That's the
Yes.
reason simply will include the protocol.
Okay.
Investigator information. The question is, who is investigator? Why do we need to have investigator information? And
Investigator means is a medical advisor through whole process, charging the whole process, right? To take care
the
the.
physician, that is a physician, clinician, most widely accepted drug.
Okay. To carry the total clinical research process. Is an investigator.
Uh, yeah, investigator is a physician, clinician, under whose special supervision drug is given to the subjects.
Okay.
Okay, then what kind of information that we include here?
Animals physiology, toxicology studies, chemistry,
That, that we include. About investigator, what
investigator.
kind of details we include? Investigator is a physician.
Yes.
We are saying investigative information also to be included.
Yes.
What, what kind of investigative information will be included?
Like protocols and the clinical trials data, everything.
Mm. Plan.
Yes,
We have
it's a
fact
protocol.
clinical.
Yeah.
So, investigative details, investigator qualification, investigator experience, investigator therapeutic area that he specialized with will include all these details about investigator.
Okay.
So, why do you want like why do it FDA require this information?
Because to approve the clinical trials, the clinical research and everything.
So FDA should know whether qualified person is conducting research,
Okay.
whether he is medically qualified or not, whether he is enough experienced, enough trained.
Okay.
All these will be in the.
FDA. Okay.
So investigator protocol, like investigator details and protocol, investigator brochure. So what is IB here? IB stands for investigator brochure. It consists of all the information about the product.
Okay.
Then why do we call it as investigational brochure? Why call it as investigator brochure?
It includes all information.
All information about the product, not the
Yes.
investigator. Okay.
Yes. Okay.
It is a document which is given to the investigator for their purpose.
Okay.
Investigator means investigators should know about the product before they start clinical studies. With this document, sponsor will be providing the information to investigator.
Okay.
Explain product, explaining the surgery, tell, develop, and give protocol to this investigator.
Okay.
CRA also say. Okay. This is clear, right,
Yeah.
Nandini?
Yeah. Yes, ma'am. Yes.
So no need of types of the IND, so which is not important.
Okay.
So after completion of IND application, we'll start with clinical study.
Okay.
Safety
And
is the same.
okay.
New.
New drugs.
So clinical trial will be discussed in types, designs, phases.
Okay.
So types of the clinical studies, let's say treatment trials, preventional studies, preventional, screening, diagnostic. So what are the treatment? Whenever we find better ways, better drugs, better devices, better test, better process to treat the particular disease, it is
Yes.
called treatment trial. So whenever we find better ways
Better.
to treat the disease.
Okay.
What is prevention?
To cure the diseases.
What are preventional?
To
Example,
diagnose the diseases.
preventional example, vaccinations, contraceptives.
Okay.
Whenever we find better ways to prevent the disease, who already had the disease or disease from returning.
Okay.
What are the diagnostic? Nandini, here you need to know the general difference between screening and diagnosis.
Okay.
So screening is nothing but general screening of the subject. Diagnostic,
Like X-rays, we should
hmm,
chemical.
knowing more about the disease or condition, health condition or symptom signs, knowing more about them, in-depth knowledge of the product or like
Okay.
disease.
Okay.
That is diagnosis. General screening for the disease is screening. So clinical trials not only for the drugs, clinical trials for the vaccine, devices,
Okay.
medicine.
Surgical procedures, everything. Yeah.
Hmm, correct. So types of the clinical studies. We have designs of the clinical studies. So we have four designs. Design one based on the comparison. Design one
Okay.
based on comparison.
Comparisons. Okay.
Based on comparison, we have two. Usually, new drug will be compared with a standard drug.
Okay.
In case if there is no drug in the market for that particular disease, then so whenever the new drug is compared with the standard drug, it is called active controlled clinical trial. If
Okay.
active drug is not there, like COVID-like situation,
Yeah.
the disease is new disease will compare with placebo.
Placebo. Okay.
What is placebo? Yeah, what is placebo?
It's like a drug or some. It's
It's a drug, but which is with no treatment value. Placebo is a dummy pill, dummy product,
okay.
which has no treatment value, no therapeutic value. That is placebo, inactive drug.
Okay.
Okay, so based on comparison, we have two: active control, placebo control.
Okay.
So based on blinding, what is blinding? Blinding is a masking process of like not blinding means not seen.
Yes. Okay. Yes.
Masking. So what exactly we are masking? Masking the drug here. Okay. So
Okay.
single, double, triple, open based on this. So masking is a process where the clinical trial, like where the one or more parties are not aware of the treatment. One or two parties not aware of the treatment. Sometimes subject don't know which drug they are receiving. Sometimes investigator also don't know which drug they are giving to the subjects. Completely blinded.
Okay.
Drugs will be coded. Drug name will not be displayed. So they will not state so and so patient to say take so and so drug.
Okay.
So single means subject is blinded. Why, why do we need to blind the subject? If what happens if they know the drug? Let's say if I'm the subject, I'm going to enroll into clinical studies. I know that I'm taking the new drug. So there will be a lot of illusions, right? Psychological feelings, fear factor.
Yes, side effect. Okay.
Yeah, so that fear factor leads to different side effects.
Yes, okay.
We will not be able to assess those side effects are because of the drug or because of the fear factor.
Yes, okay. Because of the fear factor. Yes.
Mm, correct. So double
Blinded patient.
investigator and subject. Both the parties will be blinded. You
Okay.
know the reason why we need to blind the subject. Then what is the point of blinding investigator? So let's see what happens if they know the drug. Let's take the example. I'm the subject, I'm going to enroll into clinical studies.
Okay.
Okay. Investigator is my friend or relative. He
Okay.
is going to give good drug to me, right? Which is more effective, showing less side effects. That will be injustice to others, means to avoid bias, partiality.
Okay.
Will usually go for double blind. In triple, along with these two persons, statisticians also will be blind.
Okay.
Means they're the one they are going to draw the conclusions at the end. They are the one they are going to draw the conclusions at the end.
And okay.
So they may tamper the results, they may manipulate the results.
Yes,
That's
okay.
the reason statisticians will be blind.
Okay.
So open trial, open to all. Everyone will know, and everyone will know the product. Let's say
Mm-hmm.
generally devices, new surgical procedure, but these will go for triple.
Okay.
Okay, design three based on the randomization. Design three based on randomization.
Okay.
Randomized, unrandomized. Randomized, unrandomized.
And
So what is randomization first? Randomly assigning the subjects to the groups.
okay.
Usually we'll have two groups, treatment group, control group.
Okay.
Okay. So randomization is nothing but randomly selecting the subjects. Here and there, not in the order.
Okay.
Randomly selecting the subjects to different groups
Okay.
in the clinical trial. Okay. If you are going for the process randomization, it is called randomized clinical study. If you're not going for randomization, you are assigning in the order, it is unrandomized.
Unrandomized. Okay.
So design four based on assignment of the drug. Parallel crossover factorial. Factorial. If you have two drugs in factorial, we need to recruit four groups. Group 1 receives drug A, Group 2 receives drug B, 3 receive A and B in combination, fourth one receive neither A nor B.
Okay.
right? So that means they mainly see the combination. A and B in combination will be given third group, comparing with control group, who has given nothing.
Okay.
Crossover one after the other. First drug A, then after completion of A, they'll receive B.
Okay.
Okay.
Yeah.
So mainly they will see each and every subject get both the treatments, A and B. Third one, parallel.
Okay.
Simultaneous assignment.
Okay. Drug A and drug B. Okay.
Right. So group 2 never received drug B, drug A, and group 1 never received drug B. It's not crossover.
Yes. Okay.
Clear it? All the 4 designs.
Okay.
Can you recall once 4 designs?
Yeah. Factorial and crossover
Mm-hmm.
and parallel designs.
Other designs? This is based on assignment of the drug.
Okay.
The other designs. Based on comparison, based on blinding, based on randomization, based on assignment.
Yes. Okay.
Let's say all 4 designs will be covered in the usually protocol title.
Okay.
Just have a protocol title so that you will be able to understand how designs covered in the protocol.
Okay.
Now see the title here, especially first line of the title.
Yeah. Okay.
All the 4 designs are covered, right? Randomized,
Yes.
double-blinded,
Double-blinded
placebo control.
parallel group. Okay. Okay.
Clear it?
Yes.
Usually designs will be covered in the protocol title. Most of the protocol titles will cover it.
Okay.
After completion of clinical studies. So after completion of INDA approval, we'll start with clinical study. So clinical study phases that we have, phase 1, 2, 3, right?
Yes.
So what is phase 1? Only safety.
Yeah.
Will not see efficacy here. That's the reason we call them as non-therapeutic.
Non-therapeutic trials. Okay.
Because we will not focus on the efficacy here, only safety.
Only safety. Okay.
Yeah. So phase 1 trials designed to find out that the drug is safe rather than the drug is effective.
Okay.
So phase 1 clinical trials are the phase step in testing humans.
Humans.
Your drug may act in any way, your drug may act different way. So this is the first time that we are testing the drug.
Okay.
So mainly safe dose and pharmacology, Route of administration. 3 points are explained here, you can see.
Oh, yes.
Okay.
Yes.
So safe dose and pharmacology. So for that, they conduct these studies in 2 ways, 2 stages.
Okay.
1A1, SAD, MAD, single ascending dose studies, multiple ascending dose studies. In the single ascending, what happens? A small amount of drug will be given to small group of people initially.
Okay.
Those people will be observed for the particular period of time. Okay.
Okay.
The small group of people given small dose of drug observed for the particular period of time. During that period, if the patient did not develop any side effects, will increase the dose. That increased dose again will be tested on new group of people. That means this is a continuous process until patients start showing up intolerable side effects.
Okay.
Okay, so the stage at which the patient starts showing up adverse effects, it is called MTD.
Maximum tolerated dose.
Okay, MTD. So in MAD, what they do is multiple ascending dose studies. Multiple ascending
Okay.
will go on increasing the dose, ascending means, but multiple small doses, not one dose.
Okay.
Okay, multiple up to predetermined level because after completion of SAD only we come to MAD.
Yes. Okay.
So in SAD we know the MTD. So when we are conducting MAD, we'll know the level. Up to the level, we'll just go on giving the drug
Okay.
during this observation period of observation. So we'll collect the samples, we'll send it to lab and try to understand the kinetics and dynamics.
Okay.
So MAD is to find the safe dose. SAD is to find the safe dose. MAD is to study kinetics and dynamics.
Dynamics. Okay.
So phase two, after completion, after successfully completion of phase one, A, 1B, then they'll go with two.
Okay.
So like phase one non-therapeutic, phase two therapeutic.
Therapy.
So we have efficacy here.
Both safety and efficacy. Okay.
Okay, so to determine if the drug has an effect on the particular body, like particular disease. Also body means kinetics, dynamics, efficacy, safety.
Safety. Okay.
All will be here in the phase two. Three is confirmatory studies. Phase two clinical trial involve large group of people. So generally phase one,
Small group of people.
phase two,
More than
small also.
small
Like
also.
phase two
Yeah.
also.
Yes.
Okay. But three, we will experiment on large group of people.
Okay.
So and also they do comparison. So this is to gain more extra additional information about safety and efficacy
Yes.
to get the overall picture of benefit and risk ratio. Also, we'll compare with the new drug with standard drug. After completion of three, then what is the next step?
Approval. NDA. Okay. Next step, NDA, new drug.
New drug application approval, marketing approval that we have to get. right? So we
Yes.
have to get marketing approval. To get marketing approval, we should submit NDA, new drug application.
Okay.
The data that is gathered during preclinical and clinical both will be the part of NDA. So mainly NDA is to prove enough information to permit FDA, and the NDA will provide FDA enough information to reach safety, efficacy, safety of the product, quality of the product.
Okay.
So these are all the details that we include.
Okay.
What exactly the drug is intended to do, composition, manufacturing details, evidence for the safety and efficacy, adverse results, and controls analysis. So, drug regulatory inspectors will visit the site. They will not blindly approve your application.
Okay.
Visit the site. They visit the site and they will check. They will check
Okay.
that their record are consistent with those provided in NDA. That means whether the sponsor is providing the correct details. That means whatever the data submitted by the sponsor, is it matching with the trial data?
Okay. Trial.
They will conduct the inspections whether patients have been adequately protected.
Okay.
Okay. So second point, whether patients have been adequately protected. First point, they check the data. Second point, they check patients have been adequately protected.
Okay.
The drug regulatory authorities, after carefully reviewing the data, they will decide whether to approve or reject the drug for marketing.
Okay.
You know, even after marketing also will continue to test the product. Even after approval also, regulatory authorities will continue to monitor the product
Okay.
for unusual long-term side effects. PMS, post-marketing surveillance,
Okay.
will start immediately as and when the drug is released into market.
Okay.
This is important phase. Gathers enough evidence about the safety and efficacy of the product. And phase 4 studies mandated by the companies as well as regulatory authorities.
Okay.
Sometimes it is a shared responsibility of both the parties, sponsor as well as regulatory authorities.
Okay.
Phase 4 mainly to detect rare, unusual, long-term adverse. If such side effects detected by phase 4, in case
Okay.
if any serious adverse effect occurred during phase 4, that will result in the withdrawal of the drug.
Okay.
Regulatory authority will recall or restrict the product. So this is phase 4. So phase 1 to phase 4.
In phase 4,
Preclinical,
they'll give you drug everyone, right? Everyone, all ages.
almost.
Okay.
Let's say you have that problem, you are going to take, let's say COVID-19 you take.
Yeah.
The vaccinations, whatever released, those are all under monitoring.
Yes. Okay. Only certain age people. Okay.
So we'll have age criteria.
Okay.
You're right.
Yes.
Think you have, I will send you the notes today
Yeah.
in
Sure, ma'am.
today's recording session also. Um,
Okay.
okay. I'm going to close the session for today.
Okay. Sure, ma'am. Okay.
Okay then.
Next Saturday, same time, right? 7 o'clock.
Yeah.
Yeah.
Great.
Thank you. Thank you,
This
ma'am.
conference is no longer being recorded.
Welcome. Bye.
Thank you. Thank you, ma'am.
Welcome. Bye. Good day.
Okay. Thank you.